Chapter 16, Section 4, Regulation of the citric acid cycle. When we're looking at the citric acid cycle, all of this has to be regulated. You can't just have that cycle going through all the time, because remember, NAD is a limiting factor, just like we saw with the glycolysis. Oxaloacetate levels push-pull which direction some of these reversible reactions are going to occur, or whether or not some enzymes are going to have the correct shape to have an active site with pyruvate D, with the citrate synthetase enzyme. So, how do we regulate this? How do we keep all this moving forward? Well, we've already talked about it. It's going to be ATP, ADP levels, or NADH, NAD levels. Okay, these concentrations. If you have a high levels of ATP, low levels of ADP, well, it means you have enough ATP to keep that cell going. Why push through? If you have too high a concentration of NADH, not a very, an extremely low concentration of free NAD, yeah, these ratios, if they're high, then turn off the pyruvate dehydrogenase complex. Why take pyruvate and pump it into the mitochondria? You have enough ATP for the cell. Your limited factor of free NADs is already low. You need some of it just for glycolysis. You can't use it all up at the citric acid cycle. So shut everything down before it even starts. Let ATP levels start to drop. ADP levels go up a bit. Let NADH levels drop. NAD levels go up. Then kick off the citric acid cycle once again. Now, this is going to happen if energy demands are high. Energy demands are high, your ATP levels are going to start plummeting very quickly. PDH is going to become active, start pushing through pyruvate into the mitochondria, get this cycle started. So again, we've seen this with glycolysis, gluconeogenesis, this whole ATP, ADP levels, if it's high, ATP levels are high, you have too high of NADH, stop, 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 stop. No reason to keep going through. But if you have high levels of ADP, AMP, yes, 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 yes. What's interesting is PDH kinase is a separate protein, a separate enzyme that can regulate PDH. PDH kinase is going to phosphorylate the PDH complex itself. By phosphorylating it, turns out this phosphate group from ATP is acting as an allosteric inhibitor, shuts down the process. This is going to happen when ATP levels are high, NADH levels are high. You already have too much acetyl-CoA just waiting to be used by the citrate synthase enzyme. Flip side, you have too much NAD, ADP, too much NAD, too much pyruvate. It's going to stop PDH kinase, which means the phosphatase is going to be able to pluck off that phosphate. PDH becomes active, and then pyruvate becomes acetyl-CoA and enters into the citric acid cycle. This turns PDH off, this turns it back on. They are constantly at work, back and forth. Every time PDH does this, kinase does this, phosphatase is going to remove it, and it just keeps cycling through here faster and faster until you get this stop that comes through and says PDH kinase, hold. All PDH that's phosphorylated becomes non-phosphorylated inactive. Boom. These levels go down, these levels go up, which then turns the kinase back on or stops inhibiting it, and then you get the switch over here. As we've seen with many other metabolic pathways, the strong exergonic, the really negative delta Gs, these are going to be steps that are regulated. Because to reverse them means you're going to have to put a lot of energy back into it. Citrate synthetase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. We see that there is a change in cellular metabolisms. All we've talked about so far in this chapter have been in the textbook near perfect cells. Here's how things work. Here's the levels and how the interplay of keeping things balanced. But when it comes to cancer, when it comes to tumors, a lot of this goes out the window. We see that some of these mutations, these cancerous cells, have different parts of the citric acid cycle. These regulatory points are now lost. You can't push through downregulation of mitochondrial pyruvate carrier, so pyruvate can't get carried in. It's not transported in, thus no acetyl-CoA will be made, because the PDH complex has also been inactivated. Succinate dehydrogenase gets inactivated. This means that ATP production for that cell will be greatly diminished. That means that cell is then going to switch over more to a fermentation lactic acid production type of thing. So we see that one of the things they look for when they think you may have a tumor is they're going to be looking for lactic acid production. Do you have an overproduction of lactate? Do you have a buildup of succinate? Have a buildup of succinate, then that means in the cells, it's going to start being excreted into the tissues. So you're going to have buildup of succinate in the tissues. Well, it means your succinate dehydrogenase, which we just talked about in the last couple of slides. That's also shut down. These metabolites, oncometabolites, onco means cancer. So these cancer metabolites are going to trigger growth. Even though ATP production is greatly reduced, these cells are still going to grow. They are going to burn through a lot of the built up, amino acids, a lot of the sugar, the glycogen, stores and things like that. When you get mutations in certain genes, we see that a couple of the genes, succinate dehydrogenase and fumarase. Changes in these means that those cells now have a greater potential of becoming tumorous, meaning they're going to start going through the cancerous uncontrollable growth, uncontrollable metabolism. Yeah, I'm going to let you read on that. Not quite sure on this one. I'm not. The metabolons, you're going to get these different complexes of multiple enzymes that are forming these complexes, that are forming these clusters, and you get these clusters, complex clusters together, and there's a bunch of interplay. Malate dehydrogenase synthase, citrate synthase, aconitase, these work together. There's interplay between these. They control each other in a roundabout methods. This seems to be one of the new areas of study when it comes to cellular metabolism. I personally am not very familiar with, and that's why there's really not much in the textbook because this is still kind of new. How much is interplay, how much is the interactions, and how much is just they seem to be coexisting independently of each other, or they just happen to be processes in the same metabolic pathway. That's kind of where this is right now.